So here's a pretty basic transcription translation type of question where I'm going to ask you all about what the messenger RNA sequence is going to be, what the amino acid sequence is going to be, and maybe a few other things. You can see here I've got, it's asking you where the sense strand and the antisense strand are, and the promoter and the terminator sequences. So always start these questions by looking for the start codon. Okay? And so remember the start codon right here, AUG, on the DNA sequence is going to be ATG. So I'm doing a quick scan here. I can see an ATG there, and I see an ATG right here. So the question is, which of these is the start codon? It is not that one. So why not that one? Well, it turns out that's not ATG. Remember, for DNA sequences, we always read from 5' to 3'. So we read in this direction here, 5' to 3'. If I read in that strand, I read in this direction, from right to left. So that's my start codon right here. This is my reference. So that answers my first question here. Where is the sense strand? And where is the antisense strand, or the template strand? So the sense strand is the one where the start codon is going to be. That makes the other strand the template strand, or the antisense strand. So each of those will be about half a mark for identifying each particular strand. Next it says give the messenger RNA sequence. Okay, so the messenger RNA looks a lot like the sense strand. Don't forget that RNA has uracil, or U, instead of thymine, instead of T. So we start at the beginning, and this TAC here, so TAC, becomes 5' UAC. So we'll just continue along, and I'm going to copy the sense strand, except for wherever I see a T, I'm going to put a U. So this second codon here, TAT, becomes UAU. So keep going, do the entire sequence all the way to the end. So at this point I haven't exactly told you where to begin transcription or where to end it. So what I want you to do for the messenger RNA is copy the entire strand of DNA, or at least transcribe it. Don't just do the gene, because transcription does start before the gene and finishes after the actual gene. So question C says identify where the promoter and the terminator sequences are. So the promoter sequence, if you remember, is found before the start codon, so it's somewhere in that region. If you take a careful look here, we've got a TATA box. That's usually an indication that you have some sort of promoter. And the terminator sequence is found after the gene. I have not identified to you yet where the stop codon is. I can see it. It is right here. And we'll see that here as we do the translation part of this question. So lastly, give the amino acid sequence. So always start with the start codon. There it is, AUG. Identify all your other codons. So I'm just at this point underlining all the triplets all the way to the very end. And then for my translation, I'm going to use the genetic code. So if we look here is my AUG, and that codes for methionine. So methionine. Then I look up UAU, look it up on the genetic code, and that is tyrosine, TYR, that is the abbreviation. CCU is proline, and UAA is the stop codon. So that is my protein sequence. Very, very short protein, just 3 amino acids long, and that is how you do this question. So I wanted to finish off this lecture with one of these transcription questions with some mutations put in. So remember, this will be part of your final exam. You'll have to know how to do this for your final exam. So here's a question. As you can see, it's asking for 4 parts, and I have some different mutations that are going to happen. But before we get there, we need to do our transcription and translation to see what is going on. And so this would be a good review, so why don't we start there? So let's do the transcription and translation. So remember, the very first thing I want you to do is find that start codon. Do you see it? There it is. So remember, we are not looking here. This is not the start codon. Remember that says 3' to 5'. Our start codon is this one here in the 5' to 3' direction. So that's my start codon. Next thing we're going to do, we're going to make our messenger RNA. So the messenger RNA, we're going to copy out this strand here. So this is the sense strand. We're going to copy it out, except for we're going to change all our T's to U's. So let's do that. Here goes. So there's our messenger RNA. Next thing we're going to do is we're going to find all our codons. So I'm going to underline them all triplet at a time, starting with the start codon. Here goes. So now that I have all the codons sorted out, we can start here at the start codon and we can do our translation. So our protein sequence, I'm going to use the genetic code that's found up there in the corner. And we're going to start off. The first one is, of course, methionine. Hopefully you have that one memorized by now. UAU is tyrosine. CCU is proline. And UAA, remember what that one is. Take a look up on the chart. This is the stop codon. So I don't need to write stop. I can just finish there. So my amino acid sequence is going to be methionine, tyrosine, proline. Okay, so let's try to answer those questions. I'm going to squish this up a little bit and squish that genetic code so that we can see everything on one screen for each question. So here is the first question. It says, what would happen if the T at position 1 was changed to a C? So let's take a look. We have our transcription and translation done just as before. Now we're going to take a look particular here at the second codon. So this TAT. So what's going to happen? We're going to end with some new DNA. The new DNA is not going to be TAT. The question says it's going to be TAC. So once we have our new DNA, we can determine what our new RNA is going to be. So that's going to be a UAC instead of a TAC. Remember, all our T's get changed to U's. And then we're going to look up our new amino acid. So UAC on the genetic code is tyrosine. So we're going from a tyrosine to a tyrosine. What is that? That is, of course, a silent mutation. So that would be the answer to that question. You need to give the full answer and say that we have a new amino acid or an unchanged amino acid and what it's going to be, and also give the correct terminology. Say that this is a silent mutation. Let's take a look at Part B. Part B says, if the G at position 2 was changed to an A. So start off with our transcription and translation so that we can see what is going on. So now we are looking at, it looks like we're looking at the third codon in this particular sequence. And this particular G, notice it is on the template strand, not the sense strand. So this is going to make the whole thing just a little extra confusing, just a little extra step to sort things out. So let's take a look. What is our new DNA going to look like? Well, I'm looking at that bottom strand. The question says change the G to an A. So I'm changing this bottom strand. Instead of a GGA, instead of a GGA, it's a GAA. That means the top strand is now going to be a CTT instead of a CCT. Next, I need to figure out what the new RNA is going to be. So the CTT is now a CUU in the messenger RNA. I can take a look up on my genetic code and see what that is going to change. And our new amino acid is going to be a leucine. So I'm going from a proline to a leucine. Okay, so which one is that? That is a missense mutation. So remember to use your correct terminology. This is a missense mutation. Okay, Part C. What if the T at position 1 was changed to a G? Okay, so there's my messenger RNA and my amino acid sequence. I'm looking at codon number 2. And what does the question say again? What if the T is changed to a G? So my new DNA, I'm going from a TAT to a TAG. Got to look up the RNA and see what a TAG, convert the DNA to RNA, which is going to be a UAG. Look up on my genetic code, what's a UAG? A UAG is a stop codon. So this is going to be a nonsense mutation. So there's an example of each. We had a silent mutation, a missense mutation, and a nonsense mutation. Okay, one more question, part D. It says, what if the C at position 3 was changed to a T? Okay, so there's our messenger RNA and there's our amino acid sequence. So take a look. We have our ATG. So here we go. There's the first codon. Here's our second codon. Okay, here's our third codon. So we've got methionine, tyrosine, proline, and then over here we have our stop codon. So it turns out that this particular codon, it's not even part of the gene sequence. It's actually after the gene. So what does this mean? It means it's not going to affect the amino acid sequence. So does this have a name? No, not necessarily. It doesn't have a special name. It just means the mutation does not affect the sequence. So you may have to write something like that, an explanation of what is going on. There is a caveat. It says there that this may affect some sort of regulatory component of the gene. Remember, we have a terminatory region at the end of the gene, and this may affect some sort of regulation of the gene. But that's kind of beyond this course, so we're not really going to talk about that any further. So here's another question from the review slides that I handed out this week. And it's basically exactly like the question I just did. Again, we're looking for the sense strand, the template strand, and the sequence of the messenger RNA and the protein. So take a look at the sequence here. I'm going to look for start codons. I'll look at the top sequence, A for ATGs, and I found a start codon right there. If I look at the bottom sequence, looking along here, there is an ATG, but that is not ATG. Remember, that is GTA because we read in the 5' to 3' direction. If this was a bit more of an advanced class, I would put the stop codon in the bottom strand to make sure people are reading their DNA properly. But here's a hint for the final exam. I will always have the start codon on the top strand, 5' to 3' strand, and that will always be the sense strand. So hopefully some easy marks for you on the final exam. So there's our labels: sense strand, template strand. Next I want to do the messenger RNA. As I told you with the previous question, I want you to copy out the entire sequence of the sense strand, except you're going to change all those T's into U's. So there we go. 5' we have TT right here, becomes UU, and then there's my TATA box, TATA, it becomes UAUA, and then so on. I will copy the entire sense strand, changing all my T's into U's, and that's my messenger RNA. So next I want to start at the start codon, and I'm going to underline all of the codons, all the triplets of base pairs, just so that I can do my translation part easily. And then for my protein sequence, I'll need a genetic code. I don't have one on the slide, so you have to look one up. For this question, you will have one provided for you on the final exam, or you can load one up or print one off to bring to you to the final exam. So AUG, as always, is methionine. Always start off with methionine. ACG is threonine. GAA is glutamic acid. And then UUA is leucine. AUC is isoleucine. And then UAA, that is my stop codon right there. So STOP for stop. So there is my amino acid sequence, 5 amino acids long. I have not asked you in this question to indicate the promoter sequence, but it's somewhere there before the start codon, and the terminator sequence is somewhere there after the stop codon. So again, should be some easy marks. So here's a mutation question from the review slides. Don't forget to review what the different types of mutations are: missense, nonsense, frame shift, and silent. So here's a DNA sequence, and always start and find your start codon. So there it is, ATG. There's my start codon. Next thing I'll do with this kind of question, like before, is always identify where my codons are, so my triplets. So we have ATG, ACG, GAA, TTA, ATC, TAA, and CCG. Okay, I've got my start here, so here's my start, START for start codon. I'll circle it. And then I'm going to find my stop codons. Remember my stop codons? I have a little mini genetic code down there for you, and you'll have to take a look and convert all your U's into A's. But here is my stop codon right here, TAA, UAA if it was messenger RNA. So now I need to just answer the question. It says, what type of mutation is caused by deletion at position 1? So we notice here that position 1 is before the start codon. So what kind of mutation is that? It will not have any effect on any amino acids. So here's my answer. Not applicable. It is not a mutation in the gene. It may have some sort of mutation in part of the promoter or have some sort of regulatory effect, but that is beyond the scope of this course. So question number 2, what would happen if we had a substitution at position 2? So here is position 2. So in this case, we're changing our A, C, G to something else. So we could have a UCG, we could have a CCG, or we could have a GCG. So look all of those up on the genetic code. Take a little look. You can see that ACG, it codes for threonine. If we change it to a UCG, that changes it to a serine. If we change it to a CCG, that's a proline, and if we change it to a GCG, that's an alanine. So in all of those situations, you're changing that amino acid to a different amino acid. So what kind of a mutation is that? That is a missense mutation. What if we do a substitution at position 3? So we're changing it from an ACG to an AC anything else. Take a look on your genetic code. If you take a look, AC and any other letter is always a threonine. So this would be a silent mutation because we are staying with the same amino acid, threonine. What about a deletion at position 4? So take a look here. A deletion at position 4 is going to take away that T, and then we're going to have a TAA. So that type of mutation is called a frame shift. In this particular frame shift, we're going to have a premature stop codon. So this is a frame shift that also leads to a nonsense mutation. So make sure that you can interpret these kind of questions, because I will have a big question on the test that's dealing with translation, transcription, and mutations, and it's going to be worth a significant portion of your final exam.